Thank you for inviting us to present ATLASea's updates for most of 2025 and presenting a bit the projects for 2026. I noticed that Patrick is on the call as well, so he can jump in if I forget anything. Patrick is co-director of the ATLASea program with me. Okay, so let's see. Just as a reminder for those who haven't heard of ATLASea before, this is a kind of schematics of how the project is organized. It's mostly organized in three components. DiveSea in the middle there on the left, which is concerned with sampling. SecSea, which is concerned with sequencing, and BioDSea, which is concerned with all the bioinformatics and computational infrastructure to trace the data and analyze it. Today, I will talk a little bit more as well about the two pilot projects that are now coming into effect following two calls which we published in the course of 2025. So just to go back on DiveSea first and point out that it's DiveSea is coordinated by the National History Museum and in Paris. And is charged with the humongous task of coordinating many partners in France to launch expeditions in the French exclusive economic zone, which covers about 10 million square kilometers in all the oceans except the Arctic Ocean. So it's a very big endeavor, and to coordinate everything, we've established standard operating procedures for sampling, which are specific to all the main marine lineages, which you see here on the left, and also for photography, voucher conservation stations, habitats, and general recommendations. We're in the process of publishing all of these on protocols.io. I think some of them are already published, but at least all the ones for sample processing are available on the ATLASea website. We've started as well, beyond launching expeditions, working with the Roscoff Culture Collection, which holds one of the richest collection of marine microorganisms in the world, about 9,000 strains, mostly for 5,000 or 6,000 of them microalgae. And so ATLASea is planning on sequencing a number of those, which are of interest to people working, for example, on metagenomics, but also on species of interest for extracting molecules of interest, and so on. So many applications. So we received approximately 100 strains selected by people at the Roscoff Culture Collection. And one has been sequenced already. It's challenging because getting enough DNA from these cultures is not trivial. So it's a green algae, a unicellular green algae, Rebecca billiardi, which has been sequenced and deposited in the public domain. So ATLASea is also DiveSea has also coordinated an expedition in the Bay of Biscay. Sorry, the Channel Groundfish Survey. Sorry, in the Channel, which took place in October 25. That was to monitor, it's an Ifremer expedition that monitors fish stocks in the Channel. But there was two ATLASea teams that went successively on board to collect about 60 specimen for approximately 60 species. So it's a kind of an industrial process where huge nets are hauled on board, and people sort through them to look for species that could be of interest for ATLASea based on the fact that they haven't been sequenced yet. And so, yeah, it's a tough job to be on board these ships for sampling for ATLASea. These are a few of the specimens that were sampled for the project: cuttlefish, a couple of fish species, a sea star. But the major expedition or mission that we carried out was in Marseille. at the marine station in Andoum, which is pictured here on the left, which is really nice because it's literally on in Marseille central, but also on the sea, with access with boats for to go sampling. If we zoom in here, approximately, this is a representation of the geographic area around the south of France, which with Spain on the bottom left and Italy on the top right. On the left-hand side, the green dots you see are stations or sampling stations from an early expedition in 2023. But the ones in Marseille are the green dots that are here in the center. So these are all the places where we sampled in June this year. Also in the lake here, which is a brackish water lake, which I'll come back on it a bit later. It was a mission that we carried out in 2 stages, one in April dedicated to fish and one in June dedicated to all the other taxa. And it was really successful with almost 700 species sampled, almost 5,000 tubes stored in dry ice, in liquid nitrogen and shipped to Genoscope. We had access to lots of equipment to sample sediments with dredges on the seafloor, dive with. We even had the Tara Ocean boat to be able to access places which were not easily accessible otherwise. So it was a great human adventure. These are some of the specimens we found out of the almost 700. A non-indigenous bryozoa species, a sea urchin, which is endemic to the, not endemic, but specific to the region, a sponge, a Sidian, and this inflated file clam, which was our 1500th species sampled in the ATLASea project at the time. Just a word on taxonomy, because these are really, this is a hugely important step, obviously, for ATLASea, but also for all the other projects involved in ERGA and the Earth Biogenome project. Is while we're on site, we sample an individual from a species, it's alive, and we need to taxonomically identify it on the spot, because based on that name, we can then check goats to see if it's already been sequenced, and if so, we have to put it back in the sea while it's still alive. Otherwise, we process it for sequencing. So we need a lot of people specific for each taxa on site, and usually we call on a list of about 160 different people who have worked so far with ATLASea. Most of them are professional taxonomists, but sometimes they are just retired professionals from other fields, which just are specialists in really a specific clade, like this gentleman looking with a little magnifying glass. I think he's a medical doctor who had a practice in town and just is a world-renowned specialist of mollusks of the Brittany coast. So it's not evenly distributed. 75% of field determinations were made only by a set of 22 peoples. We sometimes use tele-identification where we're on site and we don't have someone who's a specialist on site. Sometimes people are not really sure, and so we need to reconfirm based on substantial sequencing of the sample sequencing of the genome. And sometimes we have to reassign the species to another one based on that information. Most of the taxonomists are not French, because it's specialists are sometimes far and beyond, and so we have to call on colleagues from other countries, which is great because it makes it for an international flavor on these missions. And 40% of taxonomists are women. So where are we today? We've sampled about 2,300 species from 12 or 13 locations, actually, which are listed on the side here. And as you see, the 4 main ones are places where we have been on on a specific mission, bringing in the taxonomists, the molecular biologists, the divers, the boats, and everything on one site for generally 2 weeks, sometimes 3, to sample specifically everything we can find in a specific area. So that is New Caledonia. So Nouméa is the capital there, where we went there in 2024. Dinard, where we went there in 2024 as well. Guadeloupe, where we went last year. I can't remember now if it's 24, 25. And Marseille, when we were there in 2025. And all the others are usually marine stations that contribute samples along the year, and colleagues who call in and say they'd like some species sequenced. So up to now, this dive-sea operation was coordinated by Lynn Legal, who's many of you probably know her. She's a specialist of algae and a professor at the Natural History Museum in Paris. She's recently left at the museum to take up a position as director of the Roscoff Marine Station. And so she left the coordination of the dive-sea operation to Bertrand Bedhomme, who's also a professor at the Natural History Museum and is taking over from her. And many people probably know him because he's quite well versed in everything genomics. Up to now, the 2,300 species we've sampled mainly come around the French mainland economic zone, in Guadeloupe and in New Caledonia. Just to give you a glimpse of what we're planning for 2026. So as of today, a new mission is starting in Martinique, focused on sponges with a small group of people there. And then in April, we have our big mission in Roscoff at the marine station that I just mentioned, where we're going to be there 2 weeks with lots of, again, taxonomists and equipment and people going diving and sampling with boats and so on. Just after that, literally in Marseille, we have another expedition focused on gelatinous marine species, so sea squirts and jellyfishes and so on. In June, we have a small expedition in the Thau lake, which is just next to Marseille, this brackish water lake, which is a really interesting place because it's fed with rivers from the land, but also receives seawater from the sea. And so it's a brackish water which has been really perturbed in the 70s and 80s because it was flushed with fresh seawater, clean fresh seawater from a local energy plant that was using water to cool down some equipment. And so the species there were really affected by this lowering of salinity, and this is now stopped, and the normal natural ecosystem is coming back, and we see the resurrection a little bit of many marine species that had disappeared. And so it's really interesting to go back and sample from there. Ifremer is planning the MEDITS operation in the Mediterranean, again to control fish stocks and also sample for ATLASea. In August, we're planning to go back to New Caledonia, this time on the Chesterfield Islands, about a day's sailing west of New Caledonia. Then just after that, people will also go on foot in a way, or with small boats, to explore the mangroves of New Caledonia to sample some tree species there, which are of interest for ATLASea, just at the border between, again, freshwater and marine environments. And so an expedition will also look into the Gilvinet Canyon, which is just the transition between the plateau, the continental plateau, as it goes into deep waters off the coast of Brittany in the Celtic Sea. And then at the end of the year, but this is not 100% sure, we have planning to go to the Kerguelens in the Antarctic, and again explore some mangroves in Guadeloupe, and also work with the local aquariums to sample some parasites from some of the species they have on hold there. So just to finish with the sampling, to start with the sequencing now, we were very happy with the SEXY project, which sequenced the first 100 species in September 2025, which was a big milestone for us. And since then, they accelerated the pace, which led to another 100 being sequenced in the following 4 months. So now we're at 206 assemblies submitted at a rate of approximately 30 per month. So that is really, really exciting. This is a pie chart of the different species that have been sequenced by ATLASea so far. You see that there are biases in favor of teleosts, mollusks, and annelids, which reflect the sampling we have so far. That's not surprising. But in the coming months, we will also place a stronger focus on these cultures from the Roscoff culture collections. And depending on where we go, we might sample different taxa more abundantly. Just to point out that we try to abide to the Earth Biogenome quality standards, which is to generate reference genomes of at least 1 megabase N50 contig length and 10 megabase scaffold. It's working for most genomes except for these 3 on the left of the dotted line, which is the 1 megabase contig N50. On the y-axis, you have the log 10 of the assembly size. And so these 3 are a crustacean, a sponge, and a green algae, which have given us trouble so far, but we're working on them to improve the continuity of the assemblies. All the others are abiding to the standards. This is the sequencing is going well. Normally, by the end of 2026, we should have another 500. So that will bring the total ATLASea production to about 700 genomes. We've opened an ATLASea ambassador scheme in 2024, in December 2024, which is a way of offering the community to propose either samples that they have access to, which are marine species, or to suggest samples that we could go and collect, or beyond samples, just to propose a project that they would like to work on with marine genomes, and that they think we could get involved with. And so there's a web form where people can just describe what they have or what they need, and then we have a species selection committee that reviews it and then goes ahead if the species is not sequenced and it's within the remit of ATLASea. So since December 2024, we had 46 submissions from not just from France, but also other species or other countries that have scientists with interest in some of the species that are in the French EEZ. And so these are the 2 projects I was talking to in the introductions. They are funded by ATLASea through a call that we published, one in July and one in September. The one in July was focused on species that produce molecules of interest through their own metabolic pathways, and for which we think we can study them through the use of their genomes. So the call was successful. We selected 19 research groups in 2 axes, one in bioinformatics, one in more on the experimental side. The bioinformatics one has 7 groups and is coordinated by Anne Sigel in Rennes, who's a computational scientist, and the one in the experimental side is coordinated by Mark Koch from the Roscoff Marine Station, and will be made of 12 groups. So they're currently writing up the formal project, and is going to be reviewed in February. And the second one is now closed, also the call, and has not selected yet the groups, but we've had many proposals, and the idea here is to work on ecosystems subject to invasions, And so these two projects, once they have been formally established by the French National Research Agency, the ANR, will become fully part of the ATLASea project. So just a few words on the analyses of these early genomes we've sequenced. We made this slide about 3 months ago when we only had just over 100 genomes. So it shows on the left a phylogeny of the main lineages for which genomes are available in the NCBI. So at least 5 genomes, so just to make it manageable. And so the first histogram here are the 6,300 NCBI genomes that are of chromosome level quality. And the count here from 0 to 2,000 on this scale, you see with a huge number of hexapoda, which are butterflies, mostly from Detol, and Streptophyta, which are land plants here, and then a distribution of other lineages here. The subset of those that are marine are shown here, so just over 700 genomes, which have already been sequenced. The dark blue one are the species that are sampled by ATLASea at the time. So we were at 1,700, which we see reflects generally the lineages that have already been sequenced, but not all. So for example, we have some sampled comparatively many more crustacean that have been sequenced. And we also some algae, so Chlorophyta and Rhodophyta in vastly more higher proportion than what has already been sequenced. And so we are now into the sequencing. See, the scales are not at all identical here. We're into sequencing, and hopefully this slide shows that really focusing on marine species is really complementing well what's already sequenced in public repositories, which is heavily focused on or biased towards terrestrial species. Just to give you a few a concrete idea of what this biodiversity looks like. It's fairly unusual compared to what perhaps many people are accustomed to when looking at terrestrial biodiversity. It's actually beautiful as well. So I'm happy to share some of those pictures with you briefly, just to see what we're dealing with. And so when you look at the genomes now, just 4 slides and then I'm finished. So this is just for the 13 main lineages of marine species we're dealing with in ATLASea. On the x-axis, on the y-axis, you have the assembly length. And again, this is not on the full set of genomes we have today, but a more reduced set we had a couple of months ago when we made this slide. You see that the assembly sizes range from just a few hundred megabases for the smallest ones to slightly over 4 gigabases for a crustacean here. Sorry, an annelid here and a mollusk here. But some lineages have a really restricted range of genome sizes, such as fish here, Chordata teleostei, and others have a much wider spread, like annelids and mollusks. The GC percent, the average GC percent of the genome shows an interesting trend where all the ones you see here that are within this narrow range of between 35 and 45% GC are animals, metazoa, and the 3 that are on top here are Chlorophyta, Haptophyta, Rhodophyta, and these are algae. So there seems to be a partition here in terms of what the GC content of these genomes covers. And so when you look at chromosome numbers in the karyotypes of these assemblies, you see this range of karyotype complexity, which is not surprising. It goes from a handful of chromosome to this crab here, this arthropod, which a crustacean that has over 100 chromosomes. But There's nothing particularly extraordinary about the karyotypes of these marine species. The repeat content is also quite interesting here. We know that genomes have a huge tolerance to incorporate repeats in their sequences, and this is true for marine species as well, obviously. But just to show you this slightly, you would think that this would not be clade specific, but what we see here is that chordates, which are mainly fish here, tend to have repeats which where most species seem to have a proportion of repeats that is, say, below 35%. In annelids, it's higher with between 40 and 55%. And in mollusks, it's higher even, so more between 50 and 65%. Repeats of all sorts. So here we didn't, it's based on camera count, so we don't know what kind of repeats they are, but it's mostly transposable elements, obviously. And I think this is where I'm going to conclude, just to say that ATLASea is reaching now a kind of more mature phase, where with more than 200 genomes and a rate of about 30 new genomes per month, we are on a good track, I think, to sequence the target we set ourselves, which is about 700 by the end of 2026. It's still very difficult. There are many species for which we cannot get enough DNA or DNA of good enough quality because of contaminants, molecular contaminants, but sometimes also other species that contaminate the target species, the host species, if you like, because of symbiotic relationships. Or sometimes just extreme genomic properties like extremely high GC or very large sizes or extremely high repeat content. We're still finding it challenging to annotate these genomes as the sequences, the assemblies are produced, but we've now calibrated our annotation protocols, and we're about to release 35 protein coding gene annotations for 35 different genomes on the ATLASea portal. And when it's technically possible, we will associate them with the assemblies on the ENA repository. And we are really trying to work on collaborating across oceans, if you like, with other projects that are involved in marine genomics, because it's really important to share and to maximize competences across this particular taxons. Thank you very much. This is a picture of the ATLASea community that met at the Natural History Museum in October for our General Assembly last October. Thank you very much.